Hi, everyone. Welcome back. So we're going to get started with our next session, which is titled Clinical Trials, a panel discussion on the development pathway. And we wanted to have this conversation because we know from talking with families that sometimes trying to understand how decisions are made around clinical trials, how we land on design, how these are put together can be somewhat challenging. Maybe you don't have that much insight into it. So we convened a panel of expert developers and clinicians so that we could have a discussion around some of these decisions and then be able to have some questions as well from all of you so you can better understand, how these clinical trials, how we get to where we're at with it. So Goude from UC Davis is going to be our moderator for this session and lead us in these discussions. And so I'm going to hand it over to Erica.
All right. Hi, everybody. My name's Erika Goude. Thank you for being here. I'm the research manager at UC Davis, and I have the privilege to working alongside of all the families here, but also everybody here. And I'm not going to introduce them. I would like them to introduce themselves. So we'll start with Erik.
Hi, good morning. I'm Erik Henricson. I'm the Associate Research Director for the Neuromuscular Research Center at UC Davis and the PI co-chair of the Synergy Duchenne Natural History Study.
Hi, I am Paolo Bettica. I'm the Chief Medical Officer of the Italfarmaco group. That is the manufacturer of Goude that got recently approved for the treatment of Duchenne patients 6 years and older in the US.
I'm Kevin Flanigan. I'm the director of the Center for Gene Therapy at Nationwide Children's Hospital, where I have a laboratory and see patients in the clinic.
Hi, I'm Joanne Donovan. I'm Chief Medical Officer at Edgewise. I've been doing clinical trials for 20-odd years and in the Duchenne area for 10 years.
Hi, I'm Naz Dastgir. I'm the clinical development lead of the RGX-202 program at RegenxBIO and a pediatric neuromuscular specialist by training.
Now we're going to kind of change the setup for the families and everybody here of how some of the moderation has been, and there's going to be a couple sections, and I'm going to pause in between for you to ask questions, because with clinical trials, what happens a lot of times, have you heard this, that consenting is an ongoing process, and I feel like many times when we're doing research, we are in the middle of doing something, one of the physicians will be talking, or an industry will have an idea, and they will be contacted about a family, or a coordinator, or a PI asking a question, and we have to pause and actually address it. So we're going to change it to be a little bit more dynamic, where I'm going to ask questions for the panel, but I would like to encourage you guys to feel free to ask questions, because that is how research operates. So just raise your hand, and there will be somebody from PPMD to go ahead and help address that. So the first one is about family decision making, and I'm gonna go to Kevin. So Kevin, for patients that you see that are now having multiple trial options, and that's an avenue that has changed very fast in the Duchenne world, how do you help guide families in these decision making processes?
It's a great question, and often very long conversations in clinic just about this for those patients who have multiple choices, and even for those patients who don't have maybe multiple trial options. The first is a real discussion about what it means to take part in a trial, because it's been my experience that I think sometimes families may underestimate the impact that being in a trial has on not just their child, but on the family themselves. All of the travel, all of the obligations to do it. Some trials, traveling every a month or something to visit a place. So first is a discussion to say, well, are you up for that kind of process? And then the second one, when we have multiple trials, it really is, we get kind of into the nitty-gritty of the science a little bit. What's the investigational agent? What's the investigational approach under study, and how appropriate is it for your son? What's the likelihood to have potential benefit? And then what is your tolerance for uncertainty? Because these are trials, not therapies. So if you're going to take part in this, you have to have a tolerance to know it may not help your son. And of course, we hope every trial is going to be a positive result. We hope it's going to have benefit. So the distinction really sort of centers around those three things in my mind. Those are the major portion of the discussion we have to choose what might be right for an individual patient.
Yeah, perfect. So industry, this is for either of you that would want to touch on it or all of you. So this is a new changing environment that has really started very recently, but also for other studies. So when patients are starting newly approved drugs and commercial available therapies, such as gene therapy, how do you think about running trials to incorporate these patients? Or for clinical trials where patients were on gene therapy before, how are you guys thinking about incorporating that? And I know, Joanne, you could probably touch on that first, and then you guys could spin off on that.
Great. What I think that we've learned over the years, and maybe different than what people were thinking 15 years ago, is that there isn't going to be a single drug, a single treatment. This is going to be a cocktail of treatment, much like other rare diseases, cystic fibrosis. So how do we put these together and in a stepwise fashion? So what we're thinking about in clinical trials is to, as much as possible, include things that are already on the market. For example, we've always included exon skipping therapies in the trial. What we also need to think about is, does that affect the results of the trial? So we may include that, but then analyze those patients differently. Mm-hmm. With gene therapy, that is something clearly that has been coming along, and we knew that that was coming along. So what we at Edgewise did, what we have, as I spoke about the last session, we have a trial that includes boys on dystrophin-targeted therapies, as well as steroids, as well as not on steroids, actually. And what we also then heard the need for, well, the landscape is going to be different in a few years. How does this therapy work with gene therapy? You can think about when you're looking at combination therapy, and how does it fit in with a gene therapy treatment? Is it before? Well, that's pretty straightforward because we look at that in a trial with boys that haven't yet been on gene therapy. And then, is it after? We're not so much worried about during that. You guys are going to do that just fine, and we're not going to interfere with that. But to look at boys and enroll boys who have been on gene therapy. So we started a trial, and it was very much, I think, influenced by what we heard from the community, that there is a need for that in addition. And so we started that trial earlier this year. It is fully enrolled, actually, with the goal of understanding how the drug works on top of gene therapy. So that's how we've been thinking about that in light of the whole treatment landscape.
If I can add, it really depends very much on what is the purpose of the study.
Yeah.
So obviously, if we are talking about a registration study where, in essence, you want to you can collect all the information and the results that support the risk-benefit of your drug, you usually tend to exclude other treatment that might confound your result. But that is one study in your clinical development. Then obviously, following studies or parallel studies actually may well want to look at what happens if you combine other treatments. So it really depends on the type of study that you want to run. In a registration study, you are probably more selective. In other studies that are more similar to what is the real world, then you would be including also other treatments.
Great. Do you guys think that there's challenges that are arising now that more and more therapies are getting approved in the Duchenne world? And how do you think industry can help with that? Or even PPMD or sites themselves, as kind of we're having so many new approved therapies, actually very fast, all at the same time. It's a lot to be thrown at the community.
Well, I think we still will have to prove that the single molecule or treatment works. This still remains as a necessity. But then once that is done, I think the next most important question is related to combination. And I think this is a very important question for the very next future, because we will need to sit down and agree on how to address this key question, which is how can we combine different treatments.
Yeah, I was going to make a comment. Like, I think combination therapy is different for the different type of drugs. For example, like if your child had gene therapy with a different product, it's really difficult to get into another gene therapy trial. I think that we as a company are constantly thinking and trying to evolve to incorporate other therapies. But just as Paolo mentioned, you have to be sure that the benefit of the prior drug, like if a child was on Javinostat, we don't want to have the benefit of the Javinostat alter the benefit of our study, even in a positive way, because then we wouldn't be able to demonstrate that our drug is working in isolation. So these are the hard questions that we have to answer, and of course the safety of giving both at the same time. Some of the drugs have similar side effects, so putting the two together might actually make things worse, and then you don't know where the side effect is actually coming from.
If I can add to that, thinking outside of the typical clinical trial mode, another thing, and what we learned even looking back at like prednisone and deflazacort, for instance, is that once a medication becomes commonly used in the community, that becomes the space where longitudinal monitoring studies like natural history studies, clinical registries, those types of things become very, very important to look at what the clinical practice, how clinical practice is evolving and the degree of efficacy of single versus different treatment modalities, for instance, combination therapies that physicians are using in the community in real world practice. And so that underscores the need for continued involvement in natural history studies and registry projects.
And I have to add on to that, just my own intake on that is that natural history studies are very difficult and chart review studies are very difficult to recruit for, but I think if families know the importance that we're still collecting data to show the efficacy of these treatments, because a lot of time now on these chart review studies, we are indicating if they are on some sort of trial and something like that. So that is beneficial, I would think, for the community to think about how these chart review studies help us understand the disease more. Now, thinking about family decision making, the other thing that I want to talk about is we all know that clinical trials can be burdensome on families, on patients, on caregivers. So what I want to ask Erik in particular, because I know this is kind of his realm, but then also then I'm going to open it up to the community if they have any questions, is that what is the biggest barrier for families to participate in clinical trials, and how can we address those for the future? How can we as industry, as PPMD, as healthcare providers, help these families step by step to make this a little bit less burdensome? We know it's going to be, but how can we assist where we can?
Yeah, that's a great question, Erica. One of the, I think, I'm always amazed at the resiliency of the DMD community, especially when it comes to attending visits and participating in very highly intensive clinical trials, with the studies with weekly visits sometimes for long periods of time. We are learning more about the best outcome measures to use across different parts of the DMD population, whether it's infants or young children, older children, adults. And what we need to do, and what we are doing, is folks in the room here are doing, is working on finding ways to create outcome measures that are finally resolute enough that we don't have to measure them as frequently in clinic, but also to find measures that are related that we can measure in the community, whether it's video assessment or some sort of wearable technology, tracking steps or walking velocity, things like that. So using a combination of less frequent clinic visits and more frequent community-based measurement may give us the same ability to answer clinical trial questions while reducing the amount of travel and life disruption that happens in the clinical trials that we're working on now. And they're better than they used to be, but we do still have work to do there.
So while I think what you're saying, Erik, is actually very important and is probably the future, In the meantime, I think what is most important is that, firstly, while the people are writing the protocol, you need really to ask yourself how frequently you really need to see patient. And because obviously we tend to exceed every time, but we really need to think about that. The second thing is there are some assessment, and I think COVID has been a big lesson for everybody, that actually can be done without actually needing the patient to actually travel to the site. Just to give you an example, some safety assessment lab tests can be done and organized at the home of the patients. The last thing is really try and ensure that all the logistic of traveling is actually taken care of. That is actually very important. Not so organizing travels, reimbursement, all that. The more we can do on that side, the better for.
And I think that's important. That sometimes, we're always looking at the big picture, which we should, right? But sometimes, what I found in the lab that's been very beneficial, and we've had some partners that have, heard us, is that for these really long visits, to have snacks, to have bottled water. It's some toys for the kids. Sometimes it's those little simple things that just soften these really long visits. Some things we can't get away from, right? Right now in our trial design with the long functional visits, or if there's a child's having a muscle biopsy, to coordinate with a child life specialist to be there. So I think that sometimes I would encourage industry partners and sites themselves and PPMD centers to think about how can you make this a little bit easier for families. So I'm going to shift gears. Does any family members or community members want to talk about family decision making and kind of questions about that? So I'm going to go ahead and talk about placebo use. So what I'm talking about right now is why is there a regulatory emphasis on the use of placebo control groups and their approach to decrease placebo exposure? So this is all about placebo. If you guys can explain the importance of placebo, and then how could we reduce that in the future? That's a loaded one.
I have strong opinions on that. I personally think for gene therapy, it's almost impossible to placebo.
I have that as a question for you too.
Yeah. I think that we've seen in every trial, any child who's receiving gene therapy will have some changes in their, not necessarily a safety signal, but some change in their labs that will give it away. Clinically, they look like they're on the verge of getting sick with nausea, vomiting, fatigue. How can you blind that? I don't know the full details of patients who come into our trials because I'm kind of watching from a distance and only seeing numbers, unfortunately. I can't be as hands-on as I used to be as a clinician. But we had a story at one of our sites where one of the moms apparently had a child in another trial that was placebo control and knew that her child didn't receive drug and was convinced that her other child would be antibody negative because of it. And she was right.
Yeah.
So if you can't even blind parents, you can't blind clinicians, what's the point? And also the duration of placebo to actually establish benefit of your drug compared to placebo is probably beyond 1 year. So why would you withdraw care from another child for more than 1 year to really establish benefit of your drug? I personally think it's unethical.
And I think that explains the importance, again, to natural history studies and these chart review studies, because if we want to move the model of placebo, we need more natural history data to compare. And especially now, I would think with the new treatments evolving and getting approved, we would need, I don't know if you have a chart review study coming up in the future, but for other things, we would need that to be able to have that comparison. I don't know if my assumption's correct. You guys are the experts there.
No, please.
No, I have to second that. I think there has always been a message from FDA that there are instances where use of historical controls is appropriate and desirable. And I think that has been expanding of late, and there are pluses and minuses to using historical controls and making sure that the data that you're using from from whichever natural history study you've chosen is representative of and similar enough to the patient population in the clinical trial. But the other thing is from a safety evaluation standpoint. It's historical controls for safety evaluation is a little more tricky. It's possible to do, but in that case, having well-defined standards for comparison is important. And I think you've got the UDP project. You're doing the same kind of thing.
If I can, I think at the moment, especially from the regulator, there is the concern that the tests that we are using are potentially biased if the patient knows which treatment they are on. I think until we actually provide data showing that this is actually not the case, that the tests are actually solid and cannot be biased, this will always remain a problem. So the more we can do to reassure the regulators that if you run an NSAA, I mean, even if you know that you are on the active treatment, you cannot force yourself to do something that you cannot do. Until we have that answer, I think it's going to be a difficult battle.
I knew you guys would be eager to talk about this one.
So I want to agree with many things that have been said, but still put in a plug for the value of placebo. And I think it depends on what is being tested largely. So I agree very much with Naz that it is essentially impossible to blind families in systemic gene therapy delivery. But I'll point out that, it depends on what the agent is being tested, what the requirements are, for example, for steroid stabilization. There is no question that if you put patients who are steroid naive on steroids, they're going to have a benefit. And if your trial design is such that you're requiring that at initiation, it's really critical to tease out what might just be a steroid effect from non-steroidal effect. And I'll agree with Paolo, too, about these effort-dependent studies are liable to parental hopes, and even, I hate to, I'm aware in the old days when 6-minute walk was a thing, coaching to children to say, either to get into a trial under a certain number or to do better on there's a susceptibility to that. So it depends on, I think, what the question is being tested to see appropriateness. But there are certainly valid reasons to consider it. I think one of the questions that's important for parents to think about, for companies to think about in their design and trialists to think about in performing it is the placebo program, proposed placebo, ethical in its time, in its timing? If we're delaying onset of a drug that we really don't know in a young group of patients for 6 months, that might be an ethical decision as opposed to other settings. And I'm talking in really general terms here because we kind of have to.
I know we have a family, or I don't know if a family member, a community member, I should say. Go ahead and ask your question.
Good morning. So in line with kind of the placebo effect there, how would you guide a parent? So we have an itty bitty, he turns 1 tomorrow, Connor. So he's not eligible yet for really any treatment other than steroids. So as he becomes age eligible, how would you all guide a parent to either go what's commercially available when he becomes age eligible, and I'll say the for sure thing and a good option, versus maybe going the trial route for a better Gen 2, Gen 3 option? And then also, is there a concern that the Gen 2 or Gen 3 options may slow down because more parents are gravitating toward what's available and what they know they're getting?
Great question.
I'm very sorry. I had real trouble hearing it up there.
So what he's asking, it's okay, I can help. He's asking how, as a parent, do you, and I'm going to summarize, so if I get it wrong, you just correct me. But basically, as a parent, when you have these approved drugs and then you have these study treatments, how do you weigh your options and speak to clinicians to ask the appropriate questions?
How do I weigh the chance or weigh the decision of going with what I know is available and a good product versus...
How does he go with what's available to a study drug right now?
That's the fundamental question. And I do apologize, just so you know, getting old and everything, but the sound is really terrible up here. We can barely hear each other.
Luckily, I'm very loud, and
Next
I'll just
year,
yell it to you guys.
we're putting in a plug for PPMD to put some monitors here, like we're Taylor Swift concert or something. That's the fundamental question, the bird in the hand versus maybe two in the bush question. I really feel for every parent who has to make that decision, who's faced with those choices. The truth is, I won't speak for everybody. I never have an answer to that, right? Except sitting down and saying, what are your hopes and expectations about a trial drug? It's a detailed conversation. What's the likelihood of this trial drug based on preclinical data or early studies having the meaningful impact you might expect when you're looking at gene therapies, for example? Not to compare one, we can't yet compare one against another, but how do you make those decisions? How do you interpret what's out there for preclinical data to say, could this new one be better, or is it likely to be significantly worse, right? It's a long conversation. There is no one answer to that, and that's where your sort of tolerance for uncertainty, I think, comes into it as well. I encourage you to have a long conversation with your provider about these to figure out what's the right fit for you. I've never met a parent who's made a dichotomous decision like that who feels, I'm absolutely sure I made the right decision some years later. There's always some uncertainty with it. But once you've made it and going down that path fully is, I think, the best thing for you and your child. Once the decision is made, don't look back too much on it, because you're making decisions with imperfect information. If we knew which was better, we wouldn't be in this situation.
And I would like to add that the challenge, too, for the community and for family members is that it's exciting because we keep having new things come up each time. So you might have a conversation with your provider, and then 6 months later or a year later, there's something else that's coming, and when you're thinking about it, so it's okay to ask questions and to ask it again, because all of these panel members right here know that it keeps changing and data keeps coming out, so it's okay to revisit the question and feel comfortable doing that.
I just wanted to put a plug that we did open up a new arm in our study where your child would be eligible for in the 1 to less than 4-year-old group. But I have to say, again, it's a study. I'm studying it, the results of the study. You're studying the results of the study with me as you see how your child would potentially improve. So especially during a phase 1-2 portion, the ultimate goal is safety, to make sure that the drug is safe in your child. This is an unknown, and we're both embarking on this together, but please know that I think everybody on this stage could say, all of us in our companies, and in our practice, do everything we can to make sure that your child is safe to the best of our abilities, like to make sure that the right labs and the monitoring is done to make sure that everything is good for your child and safe first and foremost. But the efficacy portion is the part that we just have to learn together.
We'll have another question right here, and then make sure you guys get close to the mic, because...
All right, thank you, Erica, and you gave me the perfect lead into my question when you kind of ask this, revisiting some of these questions. And this is a comment or a question to Erik and to, Kathy in the room. In 2017, we did the formal exercise of sitting down in a workshop and really mapping out what are the unique burdens that we can identify in these pediatric Duchenne clinical trials. We mapped it out into 4 domains, right? It was psychosocial, emotional, financial, and then kind of logistical when we think about how these trials are designed. And I think there was a consensus paper that was published. I'm just wondering if it might be time to revisit that exercise to say how has this burden landscape changed in the advent of approved therapies or the maybe anticipation of longer trials, right? How do we really kind of formally look at that and map it out for educational purposes and decision-making purposes?
And I think so the answer is yes. The simple answer is that is something that's on the agenda for us to revisit and modify, because I think that that's something that's challenging for all of us, but it's a great challenge, and I like challenges, is that we keep having to revisit these standards that we set up that were phenomenal as a community, because guess what? Care is getting better, new research studies, we're now doing travel accommodations better and things like that, but you're right, we can't settle. We got to just keep pushing the envelope and see how we can do more. So that is, I'm glad you brought it up because that is something we will be revisiting.
Yeah, I do think about this as a continuous process that we need to go through, because every advancement that we achieve is going to also create new challenges. And this is the group together that's going to help overcome those challenges. So we need to maintain a regular open dialogue about exactly that.
Yeah, and I think one of the primary purposes for that exercise a couple of years ago was to ensure that regulators also understood what families are being asked to do, what companies are being asked to do, and I think it's just a terrific time to revisit for that purpose as well.
Thank you. I'm going to ask one question to Paolo and then open it back up. You guys don't keep standing because it's just one question, because I want to make sure this one is asked because I wrote it down specifically for him. Because this is about length of study. So if you guys are unaware, Italfarmaco had an 18-month long study for Javinostat, and I kind of want him to explain to the community of the importance of the duration of that study and why it was successful.
So actually, the duration of the study was one of the key questions when we actually wrote the protocol. And obviously, we discussed that with the community, we discussed that with the physician, and there was obviously concern, especially given the fact that it was a placebo-controlled study, that the length of the study could actually be a problem. Really taking into consideration all the information that we received, at the end we decided that the 18 months was actually an appropriate period. And if you look at the results which are now published on the Lancet Neurology, you can see that it was actually a good decision, because not for all the endpoints, but certainly for the primary endpoint, the effect at 18 months was actually much larger than what we had seen at 12 months. So I think it was actually a very important decision. If I have to single out the most important decision of the study, this actually might be the single one. And because at the end of the day, especially given the if you're on population that we selected, which was a population with some decline but not a dramatic decline, unless you actually follow up for 18 months, it's actually very difficult to see an effect.
This is a challenging question for you before we move to the audience, but are you thinking about opening up a study for younger?
So actually, there are two studies which are either already ongoing or they will soon start. These are actually part of what is called the Pediatric Investigational Plan for Europe. One is actually a phase 3 efficacy study in non-ambulant pediatric patients, so 9 to 18. And the second one is actually a pharmacokinetic and safety study in boys from 2 to less than 6 years of age.
Wonderful.
And this was done, I think it's starting in September.
Thank you.
My question is there one place that we can go look and say, our son has a duplication of 2? So your product may be red, he's not qualified. Your product may be yellow, he is at more risk. Your product may be green, you've seen some benefit. To where we don't have to filter through everybody's specific inclusions, exclusions, age, ambulatory. Is there a go, no-go page?
Do you want?
So the short answer is no. but the first thing, I just want to make sure everybody in the audience is aware of the clinicaltrials.gov. I think everyone is and should be. You do have to filter through individual exclusionary criteria there. I don't know of any centralized site that lists all of these as it'd be nice if there was a matrix for you to do it. And if PPMD has one, I'm unaware of it. Maybe it's something they could consider putting together, I guess.
Yeah. Erik might want to address that.
Oh.
I was just going to hop in to say, if you have those questions, please reach out through PPMD for you, the one-on-ones. Our genetic counselors do sit down with families all the time and say, based on your inclusion, we can walk through what some of those trials are with you, so that way you're not having to do some of that work. Because clinicaltrials.gov is great, but it is tough if you are not experienced with it. And so it's good to sit down with your clinician or your care team, but yeah, we can certainly help guide you through that. So we have resources that we can hopefully connect you with.
Thank you.
Sorry, Erik, I couldn't hear what you were signaling there. So. Go ahead.
So it sounds like the consensus is that a longer, at least an 18-month trial is what's needed, and you've shown that very nicely. So then I'm coming back to placebo. We don't want children to be on placebo for 18 months, for a long time. So how long should placebo be? And what is the value of a very short placebo? And how do we overcome that to help the community, but also make sure that clinical trial data is robust enough when it goes in front of the regulators?
Great question.
Yeah, so I like that question. There are actually a number, first of all, the longer clinical trials, I think we're going to see that becomes more the norm, because it gives us a chance to really see a separation between groups and ensure our chances that we don't miss that when it's really happening. But from a standpoint of duration of placebo, there are challenges in implementing some of the designs that involve just crossover groups where people may be assigned to receive a therapy at, say, baseline or 6 months later, and they may have a placebo up to that point, and you don't know whether you're starting the therapy today or whether you're starting it in 6 months, and everybody's evaluated a year or 18 months. So there are alternate designs that account for some of that, and again, there are challenges to implementing those somewhat, but it's not, but it's something that is it's under discussion frequently, so it's not necessarily an all or nothing kind of thing.
Yeah. One of the issues for placebo-controlled is particularly safety, what the FDA wants to see. And people are influenced. You hear about the placebo effect, but if you are told in the ICF, the informed consent, that the side effects are hangnails and headaches, then almost certainly both the placebo group and the active treatment group will have more of that than you would have ordinarily seen. So the placebo group really helps you understand whether or not something that you may think is related is actually related, or is suggested, basically.
We have some questions on this side.
If I can just
yes.
add one point. So related to the duration of the study, I think the other important point to keep under consideration is really the inclusion and exclusion criteria that you select, because the duration of the study of 18 months, as we did in our study, we thought that it was appropriate, given the fact that if you're on patient population being selected was actually going to decline, have some decline in 18 months, but not a dramatic decline. So that the two things together then also supported the possibility of actually using a placebo.
First of all, thank you for your effort with our kids, because it's very important. Just for the gene therapy, I do want to mention that, and I haven't heard through most of the panels throughout the 3 days, when you're doing a gene therapy, you actually have to pass the immunology test for the vector, so you cannot have high antibodies, and it's a very common virus to have. So as your kids age and they go to school, it's pretty common that they get the virus that they use. So when parents make the decision whether or not to do gene therapy, whether or not it's Sarepta's that's on the market or the trials, even if you get accepted based on the criteria of age, deletion, etc., you still have to pass the immune test, and that's not emphasized enough.
That's not, I missed the last.
It's not emphasized enough. I don't think it's really been mentioned here.
So for the panel that didn't hear, is the importance of consent. This is how I would handle it as a coordinator and a PI, is the importance of consenting and explaining to the family that you can meet all the inclusion-exclusion criteria, but then you can get to a lab test and be positive for antibody, and it could be very devastating, and I do think it's something important that maybe we could figure out a way to lessen that burden up front before all of everything that's going on.
So, it's a great point, and we often get asked, hey, could you screen us for antibodies today in anticipation of a year from now, when my child particularly might have access to a test. There are many clinics that have access, and some companies that have access to pre-screening programs for those. But the caution to it always is that decision, if we do that today, it doesn't mean you're in for sure. You have to rescreen, and you could seroconvert, the term we use for requiring antibodies. I do think if there was widespread availability, I can imagine a way it would take anxiety away from a lot of parents in removing certain things from the table, right? But we don't really have great programs to do that in our clinic. In our clinic, our practice has been to pre-screen if we were going to invite a patient to come before we do the whole screening process. They get tested a second time after the informed consent. We have a separate research protocol for pre-screening individuals to try to say, before we have you come and do all the other labs, do all the other stuff, let's find out if you could be eligible on the basis of this.
It was more of a statement just for the crowd. So when you're making the decision of, should I do the test now or should I wait? It's a gamble, because we were told once our child got the school age, it's like 50% or higher that they will fail. So you're risking, do I take the gene therapy now and hope that we get accepted, or you wait 2 years from now and now your son fails possibly? So I just wanted to let the public know that.
Thank you. A question?
Hello. So my question is about the advances in artificial intelligence and machine learning, and how that will help with experimental designs, maybe streamline experimental designs. Thank you.
Yeah, so if you guys didn't hear, it's about kind of like artificial intelligence and all of this new state-of-the-art. I will let Erik, because he does have a project, talk a little bit about how that is. And we've seen other groups also present, and you're right, is that with technology advancing, it's really an exciting time. You know, can we do quality of life assessments better? There's a lot of things that I think you are correct, is how do we advance and push the bar even up?
That's a great question, and I think that the first place that we're really going to see utilization of machine learning-based techniques, AI, deep learning, is actually a little bit outside of the clinical trials realm, but looking more globally at large data sets, clinical data sets combined with biomarker repository data sets, combined with other information about clinical care, those kinds of things, where those types of tools enable us to develop associations and find patterns that we simply don't have the ability to find in using more basic biostatistics. And so what that will teach us is more about the variability that we see typically in patients with DMD, but perhaps find mechanisms that are causative for those things. So I think that will happen quietly in the background, less in terms of clinical trial design, but more in the biology and discovery part.
I have one question before we move to the next. Oh, did you want to add on? Apologies. And then we'll open up more. So the one question, because I'm going to talk about outcome measure and design. We're out of time. Oops. Well, I'm going to ask the question anyways, and we're going to keep going because it's in lunch. So if you guys want to keep going, because these people are used to me using all your time for my purposes. So what I want to ask them is, how do you approach washout of certain drugs when patients may be fearful that a trial will fully enroll while they're washing out from the drug? So this is all about washout and starting a new treatment, starting a new commercially available. It is a topic, and how do we approach that safely, smart, ethically. It is something that families ask.
I think the most common is with steroids. Like when you change steroid doses, when you switch from one steroid to another, how long do you need to wait? So we do have a period where you need to be on stable doses of steroids, like most other trials. And then the fine tuning is, well, is it okay if you switch for something other than weight? And people have guided us to it needs to, if it's a subtle change, weekend, perhaps then it can be 3 months. But that is that's probably the most common issue in terms of washout. And we all have washouts of it needs to be the investigational drug, another investigational drug has to be completely out of the system, which we usually say 5 half-lives. It should be down to less than 5%.
Can I add, this is a special issue for the exon skipping drugs, second generation exon skipping drugs, where there's a choice of washing out, the obligation to wash out for 12 weeks or so generally from a commercial therapy, say a Teplersen, to go into the DINE study. I think there's only one ethical choice to this, and that is for investigators to insist that these studies can guarantee a screening spot to an individual patient if they complete the washout. So in fact, I know that several of the studies that have been discussed here do exactly that. They allow the investigator to say, I'm going to wash this patient out. They will, and they can guarantee a spot, a screening spot in the study. Doesn't mean you pass everything else for the screening study, but you're guaranteed a screening spot for it. And if you find yourself in that situation, you should talk to the investigator, make sure that that's part of the study.
We have one question, then we'll wrap up at like true research form. I'm going to let all of us be over here and give you guys a little extra time to answer questions because research is ongoing. You guys should always feel like you should come up and ask questions because it is a complex process.
Hi, we are now on our 3rd drug trial. The first one was the myostatin inhibitor, and that one had a placebo. There was only 25% of the boys that were placebo, and it was only a 6-month period. The last 2 drug trials that we've been in, it's a year-long placebo period, and it's 50%. Why aren't we giving our boys a better chance of getting the actual drug?
I didn't hear.
I'm sorry, couldn't hear the end of that.
She's been on 3 different studies. Do you want me to tell people they can break for lunch, or? Oh, go ahead and repeat it. Sorry.
There we go. Because you can hear from this one a little bit better.
Yeah.
Yes. She was commenting about essentially the randomization for placebo. The first trial that her family was in was, I think, 3-to-1 for the anti-myostatin. The last 2 that has been in year-long studies where it was 1-to-1. So asking why aren't we randomizing more patients to be on active versus placebo?
So we actually did randomize 2-to-1. So in our study, you had 66% chance of actually being on givinostat and 33 on placebo. This is actually done quite often. This actually increased the variability a little bit, so that means that the study needs to be a bit larger than the 1-to-1 randomization. But I think this is actually quite often used as a design, especially in rare disease.
All right. Well, thank you all. We'll be over here for a few minutes, but appreciate all the questions, and thanks to our wonderful panel. Sorry, it's a little later.